Welcome to Computational Biology. Today, we are diving into one of the most powerful statistical tools available to scientists, the chi-square test. Imagine running an experiment, gathering your data, and wondering if your results are genuinely meaningful or just a random fluke. How do you know for sure? This test acts as a mathematical detective, helping us measure the gap between what we expect to see in a perfect world and what we actually observe in reality. By the end of this lesson, you will understand exactly how to validate your biological models with confidence. Before we look at the math, let us build our intuition. Imagine you are playing a board game with a friend, and they roll a six-sided die. You expect every number to appear roughly the same amount of times. But suddenly, your friend rolls a 6 over and over again. You start to suspect the die is loaded. How do you prove it? The chi-square test does exactly this. It compares your real-world observations, the actual dice rolls, against your theoretical expectations. By measuring this gap, it tells us if the difference is just a lucky streak or if something deeper, like a rigged die, is actually going on. Now that we understand the basic idea, let us formalize it. In statistical terms, the chi-square test is a non-parametric hypothesis test. It is specifically designed for categorical variables. These are data points that fit into distinct, separate groups. Think of blood types, flower colors, or the 4 nucleotide bases in DNA. When we run this test, we start with a baseline assumption called the null hypothesis. The null hypothesis simply states that there is no significant difference between our expected model and our observed data. If our test reveals a massive gap, we reject this null hypothesis, meaning our expected model does not fit the reality of the data. You might be wondering why this specific test is so crucial for a zoology or computational biology student. The truth is, without the chi-square test, scientists would have no objective way to validate their experimental models. Geneticists use it constantly to verify if the offspring of a genetic cross actually follow classic Mendelian inheritance ratios, like the famous 3-to-1 ratio. In bioinformatics, researchers apply it to DNA sequences to see if certain nucleotide motifs appear more often than random chance would dictate, which often points to a functional protein binding site. It is even used in clinical trials to prove if a new drug genuinely outperforms a placebo. This brings us to the mathematical heart of the test. The formula you see here calculates the chi-square test statistic, represented by the Greek letter chi squared. Let us break down every piece. The sigma symbol tells us to take a sum across all our categories. For each category, O sub I is the observed frequency, the actual count you saw in your experiment. E sub I is the expected frequency, the theoretical count you predicted. We subtract the expected from the observed to find the raw difference. We square that difference and then divide by the expected frequency. Squaring ensures positive and negative errors do not cancel out, and dividing standardizes the error based on the category size. Let us see this formula in action with a classic genetics problem. Suppose we cross 2 heterozygous pea plants. Gregor Mendel's laws tell us to expect a 3-to-1 ratio of tall plants to short plants. We grow 100 plants, so we expect 75 tall and 25 short. However, we actually observe 85 tall and 15 short. We plug these numbers into our formula. For tall plants, 85 minus 75 gives a difference of positive 10. For short plants, 15 minus 25 gives negative 10. Notice how squaring these differences turns both into positive 100, preventing them from canceling each other out. Dividing by the expected values and adding them up gives us a final chi-square value of 5.33. Now that we have calculated our test statistic, we need to prove whether this number actually means anything. To do this, we must find our degrees of freedom. The degrees of freedom depend on how many categories we have. Here, we have two categories, tall plants and short plants. The formula for degrees of freedom is simply the number of categories minus 1. So, 2 minus 1 gives us 1 degree of freedom. Next, a biologist looks at a standard statistical table to find the critical threshold value for 1 degree of freedom at a standard 5% significance level. That critical value is 3.84. This is our mathematical finish line. We are ready for the final logical step. We compare our calculated chi-square value of 5.33 against the critical threshold of 3.84. Because 5.33 is strictly greater than 3.84, we have crossed the threshold of random chance. Therefore, we mathematically reject the null hypothesis that the plants follow a perfect 3-to-1 ratio. The intuition here is that the gap between our greenhouse reality and Mendel's theory is simply too large to be a lucky accident. Something else must be happening biologically. Perhaps environmental factors or genetic linkage is causing more tall plants to survive than we originally expected. Let us tackle a second example, this time from computational biology. Imagine you isolate a new viral genome that is 200 base pairs long. If the sequence is completely random, you expect all 4 nucleotides to appear equally. 25% of 200 means we expect exactly 50 of each base. But your sequencing software observes 60 adenines, 40 thymines, 70 cytosines, and 30 guanines. We set up our formula across all 4 categories. Notice the deviations. Adenine and thymine are off by 10, while cytosine and guanine are off by 20. When we square these differences and divide by the expected 50, we get terms of 2, 2, 8, and 8. Adding these yields a final chi-square score of 20. Before we conclude this second example, let us look closely at the mathematical reasoning inside those steps. Why did we square the differences? A deviation of 10 for adenine became 100, but a larger deviation of 20 for cytosine exploded into 400. This clearly shows how the squaring mechanism heavily penalizes categories that are further away from their expected values. The larger the gap between reality and expectation, the more aggressively the formula inflates the error score. Furthermore, dividing by 50 standardizes these squared differences. A deviation of 10 means something very different if you expected 50 versus if you expected 5,000. This ensures major biological anomalies are easily detected. Now we finalize our proof for the viral genome. We have 4 nucleotide categories, so our degrees of freedom will be 4 minus 1, which equals 3. The critical value for 3 degrees of freedom at a 5% significance level is 7.81. Our calculated statistic is 20. Since 20 is massively larger than 7.81, we strongly reject the hypothesis that the nucleotides are randomly distributed. In a biological context, this mathematical result is highly revealing. It suggests the viral genome has a specific non-random sequence composition. This could indicate an evolutionary adaptation, such as a high GC content, which definitely warrants further biological investigation. To truly cement this concept, let us look at the chi-square distribution curve. The horizontal axis represents the chi-square value, And the vertical axis shows the probability density. Notice the vertical dashed line? This is our critical value, the threshold we looked up in the table. The large blue area to the left represents natural random variation. If our score lands here, we fail to reject the null hypothesis. But the red tail on the right is the rejection region. When our calculated test statistic lands far out in this red zone, like our score of 20 did, the probability of this happening by mere chance is incredibly low. The diagram perfectly illustrates why we reject the expected model. We have covered a lot of ground today, so let us summarize the most important ideas. The chi-square test is an invaluable method that quantifies exactly how well your observed categorical data fits a theoretical expectation. Remember the mechanics of the formula. By squaring the differences, we prevent positive and negative errors from canceling out, while heavily penalizing large deviations. By standardizing those differences, we ensure the error is scaled relative to the expected size. Whether you are analyzing genetic crosses or studying DNA sequence motifs, this test provides a rigorous mathematical foundation. It empowers you to move beyond guessing and make confident, data-driven conclusions in your biological research.